Today we'll see the another concept in unit number 1, that is the bioinformatics. 1.2, that is the National Center for Biotechnological Information, that is the NCBI. The National Center for Biotechnology Information, that is the NCBI, it is the part of United States National Library of Medicine, that is the NLM. It is a branch of National Institute of Health, that is the NIH. The NCBI is located in Bethesda, Maryland, and was founded in 1988 through legislation sponsored by Senator Claude Pepper. The NCBI houses a series of databases relevant to the biotechnology and biomedicine, and it is an important resource for the bioinformatic tools and services. The major databases includes the Gene Bank for DNA sequences and PubMed, a bibliographic database for biomedical literature. The other databases includes the NCBI epigenomic database. All these databases, these are available online through Entry's search engine. That NCBI was directed by David Lippman, one of the original author of the BLAST sequencing alignment program and a widely respected figure in the bioinformatics. He also led in the intramural research program, including groups led by Stephen David Landsman and so on. First of all, we see what is NCBI. That NCBI is nothing but the National Center for the Biotechnology Information. It was established in 1998. It is the part of National Library of Medicine at a National Institute of the Health. The major aim is the public database and the development of the software tools for the sequence analysis and disseminate the biomedical information. Then we'll say the role of NCBI. Through NCBI, it conducts the research on fundamental biomedical problems at the molecular level using the mathematical and computational methods. It maintains the collaborations with several NIH institutes, academia, industry, and other governmental agencies. Fosters the scientific communication by sponsoring the meetings, workshops, and lecture series. Supports the training on the basic and applied research in a computational biology for postdoctoral fellows through the NIH intramural research program. Engage the members of international scientific community in informatics research and training through the scientific visitor program. Develop, distributes, supports, and coordinates access to a variety of databases and softwares for the scientific and medical communities. Develop and promotes the standards for databases, data deposition, and exchange and biological nomenclature. In NCBI, there are different sections. First of all, we will see that bookshelf. The NCBI bookshelf is the collection of freely accessible downloadable online version of selected biomedical books. The bookshelf covers a wide range of topics including the molecular biology, biochemistry, cell biology, genetics, microbiology, disease states from a molecular and cellular points of view, research methods, and virology. Some of the books are online versions of previously published books, while the others, such as a coffee break, are written and edited by NCBI staff. The bookshelf is a complement to the Entry's PubMed repository of peer 48 reviewed publication abstracts. In that bookshelf content provide established perspectives on evolving areas of study and a context in which many disparate individual pieces of reported research can be organized. Then another section, that is the BLAST basic local alignment search tool. which is given by Stephen in 1990, that BLAST is an algorithm used for calculating the sequence similarity between the biological sequences such as nucleotide sequences of DNA and amino acid sequences of proteins. It is the powerful tool for finding the sequences similar to the Curie sequence within the same organism or in a different organism. It searches the Curie sequences on NCBI database and serves and posts the results back to the person's browser in a chosen format. Input sequences to the BLAST are mostly in a FASTA or GenBank format, while output could be delivered in a variety of formats such as HTML, XML, formatting, and plain text. The HTML is the default output format for NCBI's web page. The results for the NCBI BLAST are presented in a graphical format with all the hits found, a table with sequence identifiers for the hits having a scoring related data, along with the alignments for the sequences of interest, and the hits received with analog BLAST scores for these entries. The entries is the global query cross database search system is used at NCBI for all the major databases such as nucleotide and protein sequences, protein structures, PubMed, taxonomy, complete genomes, OMIM, and several others. Entrance is both indexing and retrieval system having a data from the various sources for the biomedical research. The NCBI distributed the first version of Entrance in 1991. It's composed of nucleotide sequences from PDB and GenBank, protein sequences from Swiss Prot, translated GenBank, PIR, PRF, PDB, and associated abstracts and citations from the PubMed. It is especially designed to integrate the data from the several different sources, the databases and formats into a uniform information model and retrieval system which can efficiently retrieve that relevant references, sequences, and structures. Then another section, that is the GenBank. It has been implemented at NCBI to characterize and organize the information about the genes. It serves as a major node in the nexus of genomic maps, expression, sequence, protein function, structure, and homology data. A unique gene ID is assigned to each gene record that can be followed through a revision cycles. Gene records for known and predicted genes are established here and are demarcated by the map positions or nucleotide sequences. The gene has a several advantage 49 over its predecessor, locus link including the better integration with other databases in NCBI, broader taxonomic scope, and enhanced options for the query and retrieval provided by the entrance system. Another section is present in NCBI, that is the protein. The individual protein sequences derived from the many different resources such as NCBI reference sequence project, GenBank, PDB, and UniProt KB, Swiss Prot. Protein records are present in a different formats including FASTA and XML and are linked to the other NCBI resources. The protein provides the relevant data to the user such as genes, DNA, RNA sequences, biological pathways, expressions, and variation data and literature. It also provides a predetermined set of similar and identical proteins for the each sequence as computed by the BLAST. The structure database of NCBI contains 3D coordinate set for the experimentally determined structures in a PDB that are imported by NCBI. The conserved domain database, that is the CDD of protein, contains sequence profiles that characterize highly conserved domain within the protein sequences. It also has records from the external resources like SMART and PFAM. There is another database in a protein known as protein cluster database, which contains the set of protein sequences that are clustered according to the maximum alignment between the individual sequences as calculated by the BLAST. The last section which is present in NCBI, that is the PubChem database, which is developed by the National Center for Biotechnology, that is the NCBI. This database provides the information on the biological activities of small molecules. Search the tab for bioassay, compound and substance database. The links to reference in a PubMed and the 3 crore NCBI source databases, these are available. The detailed record for the substances, including the drug information, pharmacology, and curated literature, is also available. This PubChem database of NCBI is a public resource for the molecules and their activities against the biological assay. It is a searchable and accessible by entrance information retrieval system. Thank you.